This is Silwood Park at Ascot. It's the field station for ecologists at Imperial College London. In this box are insects just arrived from South America, and their descendants will play an important role thousands of miles away in Nigeria. They're arriving at Silwood Park because it's a quarantine station for an international organisation, one concerned especially with the protection of crops in the tropics.
Well, with biological control, what we're usually doing is trying to control a pest which is exotic in a country. It's been introduced from another country. And usually such pests get in without the natural enemies which control them in their country of origin. And what we do is we try to go to the country of origin, identify the important natural enemies, and introduce them into the new country, where hopefully they will bring the pest population down and regulate it at a low level for many generations to come.
This pest is a species of mealybug that feeds by sucking phloem contents from leaf veins of cassava.
Perhaps our biggest project at the moment involves the control of pests of cassava in Africa. Cassava is native to South America, and Africa, it's an introduced crop, where a number of pests have come in recently from South America and are causing havoc over an area about the size of Europe. It's a very large problem, and biological control has been turned to.
Cassava is a staple food for 200 million Africans who eat the pounded roots. It's their main source of carbohydrate. The mealybugs, which are a species new to science, cause distortion of the shoots, so the crop fails. The mealybug species was eventually tracked down to Paraguay, where a selection of natural enemies were collected and sent to Silwood to be screened. Of course, it's essential first to culture the mealybugs to provide their natural enemies with food and hosts.
Screening is of vital importance to any biological control project. We need to get the natural enemy in our hands so we can make sure we've got the right species and make sure it attacks the pest we're releasing it against. And then we also need to find out whether it's carrying any kinds of parasites itself, things which, like diseases or other insects, which could limit its control once it's introduced. So we want to clean it up.
In the cassava project, they've been lucky, and it looks as though this parasite, a tiny wasp, will be a successful biological control agent. But are there theoretical principles that could improve the chances of selecting a good agent? It's to the English countryside and to the cold of a November night that we must turn for some of the best clues. Nightfall sees the appearance of adults of an insect whose life history is tailor-made to give an accurate picture of its population dynamics, the winter moth. The adults emerge from their pupal cases within the soil, lying at the foot of oak trees. This is an adult female with tiny vestigial wings and a broad abdomen filled with eggs ready for fertilization. In her short life, she will never feed, but immediately begins to climb the nearest oak tree, finding her way in the dark with the help of infrared vision. Males, already emerged and on the wing, fly in attracted by female pheromones. After mating, the female carries the fertilized eggs on up to the treetop to be laid on the tips of branches ready to hatch next spring. In 1970, Professor George Varley and his colleagues had just finished a now classical study lasting over 20 years, from which they were able to build life tables for the winter moth.
He's got long white hair. Who's that fella?
Really on?
Yeah. We started in this very place in 1949, and from then, every year, we've gone through a straightforward routine, counting the insects twice a year and making sure what is happening to them so far as we can.
Their yearly routine started in April, when tiny caterpillars hatched from eggs in the treetops. The caterpillars immediately make their way to the oak buds to feed. Samples of buds were collected from the treetops, and the number that contained feeding caterpillars were counted to discover the proportion that had survived the winter. Those that managed to feed successfully eventually lower themselves to the ground to pupate. Unlucky ones fell into sampling trays, from which they too were collected and counted. The trays were of a known area, and so the number coming down from the whole tree could be estimated. Some of these caterpillars contained parasites and would soon have died, so they were recorded as having already been killed during the larval stage. The caterpillars that avoid the trays burrow into the ground to form pupae. The life cycle is completed when the adults emerge from the soil the following winter. Females usually go up the nearest tree to lay their eggs. Because the tree trunk is their only route to the canopy, it's easy to design traps to catch them as they climb. 2 traps were carefully positioned on each tree to sample from exactly a quarter of the tree's circumference. Each morning the traps were emptied and any female moths taken back to the lab for counting. From the total trapped each year, the numbers of adults that emerged to reproduce were estimated, and also by subtraction, the numbers of pupae that died in the ground during the summer. One more measurement was needed, the fecundity of the females. This was found by counting the eggs in moths dissected in the laboratory to be about 150. They found that the number of winter moth caterpillars fluctuated considerably from year to year, but only twice did they become so abundant that they defoliated the trees. One of the group who worked with George Varley is now professor at Imperial College, Mike Hassel.
What's really special about George Varley's study of the winter moth at Whiteham Wood was not just that he had 19 years of data available, but the way that he broke down the different mortalities acting through the life cycle into their separate components. Now, at this time of year, with the leaves fully out, the winter moth are already in their 3rd and later instars, and the major mortality has already occurred. And this is the mortality that occurs between egg hatch and bud burst.
Depending on whether it's a warm or cold spring, the date when oak buds open can vary by several weeks. It's critical for the survival of winter moth that the eggs hatch after the buds burst, but before the leaves are too tough to eat. If they hatch too early, the oak buds are still closed and the larvae face starvation. But all is not lost. Because they spin silken threads, some can be blown away to other oaks where there may be more advanced buds. Only if the synchrony is just right will the caterpillars find young edible leaves. In bad years, up to 95% just don't make it and die. The variation from year to year in this huge mortality accounts for the wide fluctuations in the caterpillar numbers. This winter disappearance determines the size of the population each year. It's called the key factor.
From now on, the larvae have a number of different mortalities acting upon them. Parasitism by Cyzinus is one of them. There then follows a number of other parasitoids and disease, to be followed then by the really major mortality in the soil, the pupal predation.
Predation by shrews and various beetles has been clearly identified as the density-dependent process which regulates winter moth. Like all density-dependent processes, predation exerts a negative feedback on numbers. In years when winter moth density is high, predation on the pupae is very severe, and the numbers of caterpillars next year will be reduced. But when pupae are scarce, there's proportionally less predation, so caterpillar numbers will be higher the next year. So there are 2 important mortalities. Winter disappearance, the key factor, leading to the 40-fold swing in caterpillar numbers, depending on the weather and its effect on the oak and the caterpillars, and pupal predation, acting much later, regulating the population about an equilibrium level.
George Varley was always very concerned to build population models for the winter moth, because only in that way can one really understand the contribution of the different factors affecting the life, in the life cycle of the winter moth. And what I've done is to build a series of population models that show the contribution of these different factors. This picture here shows how the population of the winter moth larvae fluctuated over the 19 years of the study. The solid line shows a population model where there is no density-dependent pupil predation at all. The pupil predation is assumed to be an average value, and the population just increases unboundedly, exponentially. Now when we include the density-dependent pupil predation, we get a very different picture. We get a population that moves smoothly to its equilibrium, which is about the average level of the winter moth population at Whiteham. Now there's something very wrong with that picture. We predicted the average level of the winter moth population, but none of the important fluctuations that are going on around that position. And this is where the winter disappearance is all important, the key factor, because it is that that determines how the populations fluctuate. And when we put in those observed values, we see just how important it is, because we have transformed the model now from one just predicting a smooth equilibrium to one where the population fluctuates almost exactly as observed about that level. Parasitoids are a major factor affecting insect populations. They make up about 10 to 15% of the million or so known insects. And the winter moth is no exception in being affected by a number of parasitoids. And one of these in particular I've looked at, which is a small tachinid fly. It's called Sizenis albicans, and that was studied as part of the life table studies at Whiteham Wood and has been included in these population models. And the really surprising thing is that the inclusion of Sizenis within the population model has virtually no effect at all. And the reason for this goes back to those very same pupal predators that affected the winter moth. Sizenis pupates inside the winter moth cocoons, and these pupal predators are not choosy. They'll eat whatever they come across. And so very heavy pupal predation is suffered by the Sizenis population as well, and this prevents the parasitoids from having a significant impact upon the winter moth population. There are two interesting twists to the winter moth story, both of which take place in Nova Scotia, where the winter moth was accidentally introduced in the 1930s and behaved in a completely different way. It increased very rapidly to cause the regular defoliation of all the trees. The second twist of the story is that Sizenis was introduced as a parasitoid from Europe into Canada to try and biologically control the winter moth to low levels. And this proved spectacularly successful, quite unlike the way that the parasitoid behaved at Whiteham Wood. What we have here is the winter moth population at its outbreak levels, and then the parasitoid was introduced, and we see this dramatic crash in the population, which then remains quite stable at these very low levels. The real difference between the Whiteham Wood population dynamics and the Nova Scotia population is pupal predation. At Whiteham Wood, a very heavy density-dependent mortality. In Nova Scotia, a slight mortality, one that allowed the winter moth population to build up and also allowed the parasitoids to be very effective after they were introduced. Now, if we know this difference between Whiteham Wood on one hand and the Canadian populations on the other, we should be able to take the Whiteham Wood model, modify it along those lines of the pupal predation, and predict the outcome in Nova Scotia. When I did this, the population dynamics showed a population that crashed, but then underwent these wide-scale oscillations, quite unlike what was observed. There's clearly something very wrong here in our understanding of how parasitism is affecting these populations. And we need to know more about the way that parasitism operates if we're to predict its impact upon the winter moth population.
It's almost impossible to do experiments with populations like the winter moth in a wood. There are just too many uncontrollable and interacting factors. The ideal experiments would involve a much simpler system. Stored flour provides an ample food supply and constant habitat for animals that can tolerate the dry, monotonous diet. Caterpillars of the flower moth Ephestia can produce heavy infestations where they ruin the grain by eating it and spinning silk. The caterpillars are attacked by an ichneumoniid wasp, Venturia. These are parthenogenetic females which lay their eggs singly, one in each host, and their stereotype behaviour is well suited to experimental observation. A wasp is attracted to a flower moth caterpillar by the smell and presence of silk. She walks about searching, and when stimulated by appropriate cues, she begins to probe into the flower with her long ovipositor. After she encounters a caterpillar and lays an egg, she withdraws the ovipositor, quickly followed by a characteristic cocking of the abdomen as she loads another egg into her ovipositor. So while watching a wasp, her behaviour can be broken down into categories, like resting, walking, probing for and encountering caterpillars, egg-laying, and cocking. A research group at Dundee University used the Ephestia venturia system to quantify relationships like those between host density and numbers of eggs laid by parasitoids. The caterpillars are put singly into the numbered cells on a grid. They're cooled first to slow down their activity. The pattern of cells with larvae added to them depends on the objectives of the particular experiment. Flour is added to all 50 of the cells. Then, to prevent the caterpillars wandering out of the cells when they warm up and begin to spin silk, netting is stretched over the grid and clamped in place. The grid is now ready for the wasps. Steve Hubbard selects from these bowls of venturia cultures one with wasps at a certain stage of maturity, all the same age and ready to lay eggs. The experiments are set up in a constant environment room, and there's a computer into which observations can be logged directly. In this experiment, a single wasp will be allowed to search one of two grids. One grid has a low density of hosts, and the other has a high density. Wasps are moved with a pooter. One is placed over each grid. When they're released, will they behave in the same way? The computer is used to keep track of the wasp's activity, logging the time spent on each category of behaviour. This wasp seems to be spending most of her time walking over the grid. She's searching, but with little success. This is the low-density grid. But here on the high-density grid, the wasp is probing and regularly finding caterpillars. And now she's encountered a host and laid an egg. More of her time is spent handling the hosts than searching for them, and of course the cocking behaviour, essential before the next egg can be laid, is part of that handling time. In this simulation of venturia behaviour, as each cell is visited, it's marked by a peg. The one yellow cell is occupied by a host, and now an egg is laid into it. The other cells are empty and shown in blue. Almost all the cells have been visited, and the one host was found. In this grid, there are 3 yellow occupied cells. In the same period of time, eggs are laid in all 3, but several empty cells have not been visited. Now here's a higher density with 9 occupied cells. The wasp spends much more time handling the caterpillars. She's found only 5 of the 9 occupied cells, and when the time is up, she's left a number of cells unvisited. At lower densities, the wasps spent more time in searching and less in handling hosts. And this can be shown as a graph, with the host density along the horizontal axis and the proportion of time spent handling up the vertical axis. This gives the classic type 2 functional response curve. So at higher densities, the numbers parasitized are limited by handling time. So far, for each grid, there's been only one parasitoid, and the hosts have been distributed randomly. But what happens when there are several parasitoids, and when hosts are not randomly distributed? In this experiment, 8 wasps are released onto a grid where the distribution of the hosts is patchy. Soon they begin to move about and search, distributed evenly across the grid. But after a time, nearly all the Venturia are probing over the cells in the lower half of the grid, in the area where a high density of cells are occupied by caterpillars. This is clear from the rings of silk in the cells. Here, the higher the density of hosts, the horizontal axis, the greater is the amount of time spent in that patch, the vertical axis. But when several parasitoids are searching a limited area, they are bound to meet each other, and some are disturbed. This mutual interference reduces the number of eggs they lay. These simple experiments with Venturia show that their search is not random, and they aggregate where there are more hosts. So how does this knowledge of Venturia behavior help to explain the success of Sizenis in Canada?
Well, the problem with the first attempts at modelling the Canadian situation was that Sizenis was assumed to search randomly over its habitat, whereas in fact what it does is concentrate its searching on high-density trees rather than low-density trees. So that percentage parasitism is density dependent from tree to tree. High-density trees suffer more mortality than low-density trees. When we put that into the population model, we get a very much better outcome. Now we have the predicted winter moth population falling very rapidly as observed and then being maintained at a stable and very low equilibrium level. This stable population is entirely due to the non-random search of the parasitoids. Their density-dependent response from tree to tree is what's regulating the population now.
We know something of the way natural enemies can regulate their prey, but how useful is this to practical biological control?
Well, when we have a biological control project to do, we're usually in a great hurry. We don't really have the time, and often not the funding, to do detailed preliminary studies on natural enemies before we introduce them against a particular pest. However, the winter moth story, I think, is an important lesson. If retrospectively we can understand how certain programs worked, and more importantly, how certain programs didn't work, then we'll have a better idea of the whole problem of biological control in the future. And there perhaps we'll begin to build up the kind of information we need to plan projects properly and to say this one will work and this one won't.